So now we're going to talk about how the DNA is actually used then to express the genes in order to form proteins. So this whole idea is called transcription, or it's called the central dogma, and it's comprised of two different parts: transcription and translation, and then a little bit of what's called post-translational modifications. So the first step is transcription, where we go from DNA and we create a message with RNA. Then that message is translated by tRNA in the ribosomes in order to make a chain or of amino acids or the primary sequence of a protein. And then there's all these post-translational modifications which then lead to like the functional, structural, quaternary structure of a protein. So, the major components that are going to be involved in this are messenger RNA, tRNA, regulatory RNAs, ribosomes, bunches of different kinds of enzymes, and then like some sort of raw materials or building blocks. So, RNA is obviously really important in the process of transcription and translation, and it's a nucleic acid just like DNA. It's a little bit different in that it is single-stranded. And then it also contains uracil instead of thiamine. And the sugar is a ribose sugar instead of a deoxyribose sugar. Right. So the types of RNA that we have are tRNA, mRNA, and rRNA. And they are going to play a role in transcription and then translation. We also have these sort of regulatory RNAs. There's all sorts of names given for them that really sort of play a role in how all of this goes down. But we're not going to go much into detail about regulatory RNAs. Just know that there's some other RNA that sort of helps in the process, right? So we transcribe the DNA to a message. And then tRNA and ribosomal RNA are down here and make up the structural components of the ribosome. And the tRNA helps to connect the amino acid sequence and associate it with the message, which then forms the structural protein. So this is what the big picture that I'm going to be walking us through. So the messenger RNA carries the DNA's message. So this happens similarly to how replication happens. We have to open up the DNA, and then an enzyme comes in and builds a polymer or a sequence of nucleic acid. And in this case, it's messenger RNA, or it's RNA rather than a DNA, but it's essentially reading the template strand in order to create the message. And the message is transcribed in threes, in little sets of triplets, so 3 nucleic acids in a row, and those are called codons. All right, so we have to unwind the piece of DNA, right? So here's our double helix of our DNA. RNA polymerase, right? So that's an enzyme that makes a polymer of RNA or a sequence of RNA. Will then bind to the beginning of the DNA or what is called a promoter region. So just basically a starting point. And then it'll go along and it'll read what's on the template and it will create the messenger RNA. So it'll build the message here, right? So then that message is going to go along and be transcribed. So the DNA is read in threes, right? So we create this triplet or this codon of messenger RNA, and every 3 or every codon associates to some specific amino acid and is going to determine what our amino acid sequence is, right? So there are a limited number of amino acids. So we'll see that some triplets will code for the same amino acid, or multiple triplets will code for the same amino acid. But we're going to build that primary structure of the protein, so this big long chain of amino acids, which then is going to form into our functional like 3-dimensional structural protein, right? So how does that message go and then create the amino acid sequence? That is through the process of translation, right? So here's our double-stranded DNA. Here is our messenger RNA. And then the tRNA's job is to correctly attach the appropriate amino acid that corresponds to the codon by pairing with it. And this happens at the ribosomes, and we'll look at that whole process of translation in a second. So these are our lists of amino acids, right? Over here, alanine, threonine, valine. So these are all of our amino acids. And these letters right here correspond to the triplet or the codon on the messenger RNA, and it tells what kind of amino acid would be placed in the sequence. So over here, you see on this side, it says the first base in the codon position, then the second, and then this would be the third. So you read it by saying, like if we wanted to look up CAC, right? The first base position would be here, C, and then we would go over and we would see A in the second position, and then we said C, so we'd follow along here, and so we end up with CAC, and that would correspond to histidine, right? Now notice there are some that are start and stop codon. So the stop codon causes the translation to stop, and then the start codon is where it always begins with an AUG. All right, another thing that we can notice is this idea of redundancy. So there's redundancy in the genetic code where a particular amino acid can be coded by more than one single codon. And in many cases, the first two nucleotides are what are required to determine what the amino acid is, and the third one doesn't really matter. And so this is the idea of wobble, almost like the message could be wobbling on the docking station, and we might still get the correct amino acid. So if we look up here, you can see that anything that starts with a C and a U is going to code for leucine. So really the third base pair is irrelevant which one is put there. And we'll see how that corresponds to the development of mutations, or for mutations to be silent in the next video. All right, so the whole process of translation, taking that message and making our sequence of amino acids. The important components in this are the messenger RNA, which we see here. We have the tRNA, which are these T-shaped things here, and then the ribosomes. And this is really where the whole, like sort of the docking station, or where it's located. So that's the big blue thing here. So the tRNA really is just a big long sequence of RNA that folds over itself, and it says it makes these hairpin loops. And as it loops around, you can see that it complementary base pairs here. So you get CGAU, CGAU. And so there is a connection, or hydrogen bonds, that are formed right here, giving it this shape. And then at the very tip here of the tRNA, there is an anticodon that is going to complementary base pair with the codon on the ribosome. So you see a U would go with the A with the U, and the C with the G. And so it would read this codon, and the corresponding amino acid from this table is connected to the end of the tRNA, right? So this is the messenger RNA code here, but it tells the tRNA when it reads this what amino acid it would have hooked on to this site. So then the tRNA is really just sort of carrying the amino acid. So as the ribosome is where all of this happens. So there are different pits in the ribosome where the tRNA can sort of move in, and then there's this dock, or this platform, where the messenger RNA sits waiting for the tRNA to come in and attach and bring the amino acids close enough together so that they can form a bond. So just a little bit of review about the difference between prokaryotic and eukaryotic ribosomes. And remember, we like to do this because talking about the different size of, or the different types of ribosomes indicates something that's a difference between eukaryotic and prokaryotic, and potentially could be a drug target. So the prokaryotic ribosome is 70S total in size, and it has a subunit, two subunits. It's got a 50S subunit, and it has a 30S subunit. The eukaryotic ribosome is 80S, right? And so it has a 60S subunit and a 40S subunit. So they're a little bit different. And remember that those subunits really just talk about their ability to sediment through. And that's why these numbers don't really correspond or add up to each other. But just remember that they're a little bit different. Okay, so this is where the translation is going to be occurring. So this is where we're going to take the messenger RNA and we are going to correspond the correct amino acid to the messenger RNA via this anticodon of the tRNA sort of reading what the message is. So what actually happens is you'll have two tRNAs come in next to each other and read adjacent triplets or codons. And because they're in here and they're close together, now we have the two amino acids that would go into the sequence close to one another, and they are going to be able to form a peptide bond. Once that bond is formed, then the tRNA will shift over and one will leave and the next will hold on to the sequence and wait for the next tRNA to come in and read the next triplet. And then it will build a peptide bond between the second and the third amino acid, and then it will shift over and the process will continue until we get to a stop codon. So this is where translation is actually terminated. So if a triplet is UAA, UAG, or UGA, it doesn't code for any amino acid, and so this is when it signals the tRNA to sort of stop and that the entire amino acid sequence is built. Then we have the whole process of doing the sort of finalization and then the post-translational modifications that happen afterwards, where we take that amino acid sequence and we fold it over itself and we add several together and we might put in some cofactors and we really get to the place where we will have the full functional quaternary structure of the protein. Now, what's really cool about bacteria is they are super efficient at doing this because the DNA is in the same area in the cell, it's just in the cytoplasm with all of the ribosomes. So multiple ribosomes can hook on to one message and create amino acid sequences all at once. So here you can actually see an electron micrograph picture of this where we have the DNA here and those little blobs right here, or sorry, this is the messenger RNA, and then these little blobs right here are your ribosomes, and you can see the amino acid sequences being formed as they move along the message. So we can create a lot of protein really quickly all at once. All right, in the next video I'll talk about mutations and what happens when there's mistakes in the message.